Hello students, my name is Shashank Shekhar and we are doing the chapter Biomolecule. So if you look at the syllabus here, see we have covered carbohydrates and proteins. The leftover part are enzymes, hormones and vitamins and nucleic acid. So we will cover the remaining part today. So let us start our today's lecture with the analysis of topics that we will cover in detail. So first of all, we will see what is enzyme. Then we will see the mechanism of enzyme action. How does an enzyme act upon a given substrate? We will see that. Further, we will move to vitamin and we will see what is nucleic acid, which is the basis of forming your DNA and RNA and all. Then we will see the chemical composition of these nucleic acids. After that, we will see what is the structure of nucleic acid and how does it helps in formation of DNA. After that, we will see structural aspects of this DNA versus RNA, means what is the difference between DNA and RNA, and what are the functionalities of DNA and RNA. Then we will move to the biological function of nucleic acid. At last, we will see what is hormones. So let us start our today's class, starting from enzymes. So you must have heard this term enzyme earlier also. So they are basically present in our body and they act as a catalyst. So what are enzymes? Enzymes are something that act as a catalyst. Now at the same time, there is a word called negative and positive catalyst. So that is a wrong terminology. Catalyst by definition or by term, it means that it is enhancing the rate of reaction. If you have to say, if you have to define something that is slowing the rate of reaction, then it would be termed as inhibitor, okay, and not catalyst. So enzymes are those substances which catalyzes a reaction, and they're generally named upon a substrate on which it operates. So there can be two ways it can be named. First, it is named after the substrate on which it is acting. The example is, if it is acting for the hydrolysis of maltose, so it is acting on maltose. So the enzyme involved in the catalyzing this hydrolysis of maltose is called as maltase. Second way, which is not used often, is the functioning way. So if it is the way of functioning, so sometimes it is named according to its functionality. Example, so if it is oxidizing something, if it is oxidizing a substance and it is simultaneously reducing something, then it is called as oxidoreductase enzyme. So since it is doing the oxidation also and it is doing the reduction also, so we call it as oxidoreductase enzyme. So the example of one such enzyme which is named after, which I have mentioned it right now, that is maltase, functions like this. So you can see here that maltose acts upon this, say our maltase acts upon this maltose to form glucose. That is the forming unit of our carbohydrate. That is, you know that maltose is made up of 2 moles of this alpha D glucose. So as we hydrolyze it, we will get the glucose back, and we are doing it fastly with the help of maltase. Clear? Now let us look at the mechanism of how this enzyme works. So here is a proposed mechanism that is being used often. So how it is proposed? Let's suppose this is your substrate and this is your enzyme. Now it works on this key lock basis system. Okay, so this is your lock, as it has a cavity here. So these are the 2 cavities. So it is just arbitrary case we are taking. This cavity shape can be different, and the different in cavity shape is the basis of specificity of an enzyme. Means enzymes are very specific in nature. They are specific to not only substrate but also pH and temperature. So they act efficiently only on a certain substance or substrate at a certain temperature at a given pH. So here we have a mechanism of the action of this enzyme, where the lock, which is called as lock key model, where the lock is the substrate and the key, which has the cavity, is the enzyme. Now they both get attached and form this complex. Now as this complex further decomposes, this gives us the product. You can see this was a substrate and now it has been converted to product, and the enzyme has been thrown back. So it acted as a catalyst here because the concentration is remained same. So we have taken one say one molecule of this enzyme, and we got that one molecule back. So it is acting as a catalyst, as its concentration is not changing, and it is facilitating the reaction. Now, similar to the action of our chemical catalysts, enzymes are said to reduce the magnitude of activation energy. So they do the same thing. Means it, from the chemical kinetics, we have seen that there is something called reaction profile, and for reaction to take place, the reaction profile goes through an activation energy, a certain activation energy, and if that energy, if the provided energy is less than the activation energy, then the reaction will not take place. So we studied there that if a catalyst is being deployed there, then the catalyst, what does it do? The catalyst reduces the amount of activation energy by some amount, and now at a lower temperature also this reaction takes place. So the enzymes do the same function. So for example, the activation energy of acid hydrolysis of sucrose is 6.22. So this is without the enzyme, the activation energy without the enzyme. But in the presence of enzyme, this energy reduces to 2.15. So you can see that how efficiently this catalyst is facilitating the reaction by decreasing the activation energy. And the catalyst that is being used for the hydrolysis of sugar is called as sucrase. So like we have for the hydrolysis of maltose, we have maltase. For the hydrolysis of sucrose, we have sucrase. So this was about enzyme. Now let us next move to vitamins. What is vitamins? So vitamins are organic compounds that are required in our diet in a small amount to perform a specific biological function for normal maintenance of optimum growth. Okay. So first of all, this vitamin is derived from the word vital plus amines. Initially, it was assumed that vitamins is nothing but the amines that are vital for our existence. But later on, it was found that vitamins are not only amines, but they are more complex structure. But the name was still kept as vitamin, but the pronunciation has been changed from vitamin to vitamin. Clear? So what are vitamins? So vitamins are small organic compounds. Vitamins are organic compounds that are present in a small amount, and they perform specific biological function for normal maintenance of optimum growth. Now, if I talk about the types of vitamins, we can have water soluble and fat soluble. So fat soluble and water soluble. What is the example of fat soluble? ADEK. So fat soluble means which are soluble in fat. So why is this soluble in fat and not soluble in water? So the one reason which you can think of is that fats are generally non-polar in nature. So these vitamins must have been non-polar in nature. For that reason, they are only soluble in fat. So they are fat soluble. And where they are stored? They are stored in liver and adipose. Okay, they are stored in liver and adipose. Where the water soluble vitamin that must have been polar in nature are soluble in water. And the groups of vitamins are B and C, like our vitamin B, except the vitamin B12. Okay, so all the vitamins that falls under the group of B and C are water soluble except vitamin B12. So they are readily excreted in urine and cannot be stored. So they are readily excreted in urine and cannot be stored in our body. Next is nucleic acid. What are nucleic acid? So every generation of every species has its ancestors in many ways. So it has been observed that the nucleus of a living cell is responsible for the transmission of inherited characters, also called heredity. And the particles in nucleus of the cell responsible for heredity are called chromosomes, which are made up of proteins and other type of biomolecules called nucleic acid. So there are mainly two types of this nucleic acid, DNA and RNA. Deoxyribonucleic acid and ribonucleic acid. So the deoxyribonucleic acid is responsible for your hereditary nature. So what it has been mentioned here is that it has been observed that we have characteristics of our ancestors. They have been passed on to us. Now, why is it so? Because the cell in the nucleus, means the cell that is present in our ancestor's body, and the nucleus inside has chromosomes. Okay, and these chromosomes are X and Y chromosomes, and they are present inside a body, and whenever the reproduction takes place, these X and Y chromosomes are transferred to a child. Now, these chromosomes are made up of protein, first of all, and the second thing is nucleic acid. So this nucleic acid is the formation of the basis for hereditary, and specifically I would say DNA. So since nucleic acid are long chain polymer of nucleotides, so they are called polynucleotides also. What it means is that you will study about the nucleotides and nucleosides later on, but this, mark my words, that these are, what are nucleic acid? Nucleic acids are long chain polymers of nucleotides, so they are called it as polynucleotides. So this is a polynucleotide, this is also a polynucleotide. Now let us see the chemical composition of nucleic acid. So complete hydrolysis of DNA or RNA yields a pentose sugar, phosphoric acid, and a nitrogen containing heterocyclic compound. See, we have seen it many times that hydrolysis is used often in biomolecule to get the basic building block. Like we use the hydrolysis for hydrolyzing disaccharides and disaccharides to get the monosaccharide unit. We use the hydrolysis of protein to get the building block, that is the amino acid. Here we are hydrolyzing our DNA or RNA, and that would give us pentose sugar. So that is giving us pentose sugar first of all. Second, it is giving us phosphoric acid. And third, it is giving us nitrogen containing heterocyclic compound. Now this pentose sugar is the only difference that is between DNA and RNA, and also the nitrogen containing heterocyclic compound has one difference that I will tell you. But let us first look at this, say our pentose sugar. You can see here that this pentose sugar is of two types here. This is beta D ribose and beta D 2-deoxyribose, which is for DNA. This is for DNA and this is for RNA. Now what is the difference in the structure? You can see here that at the second position there is a OH group at the alpha beta position, whereas in this case it is present as H. So what we have done, we have kind of deoxygenated this thing, and therefore it is called as beta D2. At 2 position we have deoxy group ribose. Okay, so this is the name. So the same thing written here, the sugar moiety is beta D deoxyribose, whereas in RNA it is beta D ribose. Clear? Now let us look at the overall structure of nucleic acid. So for that we will look at the chemical composition. The next thing is the nucleic acids that are present. So I have told you that it has a phosphate ring, it has a pentose sugar and a nucleic acid. Now what is the say our nucleic acids that are present? So DNA contains adenine, guanine, cytosine and thymine. So this is the thing. RNA also contain the 4 bases. The first means they also are made containing 4 bases, but all the 4 bases are not same. The first 3 bases are same, means adenine, guanine and cytosine are same, but the fourth one is uracil, means the 4 bases that are present in our RNA are adenine, I am representing it with code word, guanine, cytosine and uracil, AGCU, where in this case we have AGCT. Got it? Now next thing, these are the structures of our, say, nucleic acid. So this is adenine, this is adenine, this is guanine, this is cytosine, thymine and uracil. So these are different sets of base. So they are called nucleic bases, and these are different sets of bases. Okay, clear? So they both are of different sets. Next, let us see at the structure of nucleic acid. So how is a unit formed? A unit formed by the attachment of a base at first position. So there is a base at the first position, and this is called as nucleoside without any phosphate group. Now in nucleoside, the sugar carbon are numbered as 1 dash, 2 dash, 3 dash, etc. in the order to distinguish this form this form the base. While in nucleoside is linked in phosphoric acid at 5th position. So when we attach our nucleoside at 5th position with a phosphoric acid, which is PO3, then we get a nucleotide. Okay, then we get a nucleotide, and this phosphorodiester linkage between 5 dash and 3 dash. So these nucleotides, what are these nucleotides? The nucleotides are bonded together. How? By phosphodiester bond linkage between 5 dash and 3 dash. So we will see the linkage, how does this linkage take place. So this is 5 dash and this is the 3 dash. Okay, so here is an example. So what we have taken, first we have converted our say, oh, sorry for the small image, this is 5 dash 3 dash. Of course this 2 will react, this 2 phosphate containing nucleotides will react, and you can see that fifth position is being attached to the third position with the help of a phosphodiester bond. So this is a phosphodiester bond. Let me draw it bigger for you. Here we have here O, CH2. So here we have the third position. Here we have the third position, and here we have the fifth position. So this is 1, 2, 3, 4, and this is fifth. And this is 1, 2, and 3. So 5, 3 phosphodiester bond has been formed. Now such phosphodiester bond has been formed repeatedly. Okay, these are formed repeatedly, and now this structure takes formation of this structure take place. Now one question has been asked more often, that suppose you are given the code for one of the strain and you are asked to give the code for another strain. I mean that we have seen that adenine, guanine, cytosine, and all of this have a say code. So adenine has A, adenine, then guanine. It has G, cytosine. It has C, and thiamine. The structure you have seen right now, that two alpha helix being coiled together, this is the structure of DNA. While the single one which do not have any say side chain is called as RNA. Coming back to this, suppose you are given one DNA and you are given the code of the DNA like A, G, T, A, C, T, A, and you are told to write the next code, means the code of the next thread. Suppose the code of the red thread is given, the code of this red thread is given, and you are asked to find the code for the blue code, for the blue thread. Then how you will find it? So for that you can just look here. If you look at, there is a hydrogen bonding. So 3 hydrogen bond between C and G has been formed, and between A and T 2 hydrogen bonds are formed. So in this way these two strands are held together. Okay, so if G is present on one of the strand, then it is necessary then C, a corresponding C will be present on the either side. Similarly for A there will be a T, and that is followed for the converse chronicle order also. So if you are asked to give the say substitute for first strand, and you are asked to give the code for the next added second strand, then you will give it as, so A turns to T, similarly T turns to A, and G turns to C, and C turns to G. So A gives us A, G, C, T. Sorry for this. Yeah, we have A, T, and C, G. Yeah, so therefore A gives us T, G gives us C. So I am writing is the corresponding code in the downside. Again T replaces A, then A replaces T, C with G, T with A, and A with T. So this is the corresponding second thread. And why is it so? So that because we have to form the hydrogen bonding, and that is formed between G and C and A and T. And one more thing, there are 3 hydrogen bonds between C and G, and there are 2 hydrogen bonds between A and T. So this should be kept in mind. Next, this is the structural aspect of DNA and RNA. So as I have told you earlier, that in DNA we have the hydrogen bonding to stabilize a double helical coiled structure. So this 2 double helical structure is in form, and you can see here there is a hydrogen bonding in each of this. Whereas in this case there is a single strand. Okay, so we have a single strand in RNA and double strand in DNA. Next difference was that the difference in the third position, we have a hydrogen, that is 2-deoxyribo, whereas in this case we have hydroxy. For say our DNA, we have thiamine cytosine, guanine, and adenine. Whereas for our RNA, we have uracil, and all three are same: cytosine, guanine, and adenine. So the only difference is that DNA contain only thiamine bases plus these three bases, whereas RNA contain uracil bases plus these three bases. Okay. The overall nucleoside structure was looking like this. If you remember it, is the 14 page, sorry. Yeah. So here we have the complete structure as I have drawn it here. So let me draw it again for you. So we have a sugar here. So I'm drawing it for DNA. So this will be pentose sugar that has 2 deoxy group here, that is H is present on both sides. Now at first position, this is the first position, we have a base. So 2 bases are present here. Next, at third position, we have the phosphor group. Got it? And this is being linked to another ring that is at the 5th position, CH2. Here also it will have a base similar to the previous group. And at CH2 again, for this CH2 again, we will have our phosphor bond, which will be instead attached to another. Before this will be P, O minus, and this will be again attached to another one. So in this way, this will be elongated. Now, let us look upon hormones. Before that, let us look upon a difference upon RNA and DNA in the theoretical form, that was of the imaginary image form. So sugar backbone. So ribose contain hydroxyl group at second position. Deoxyribose lacks this hydroxyl group. The bases that are present are adenine, guanine, cytosine, uracil. Whereas in DNA we have adenine, guanine, cytosine, and thiamine. Structure. Talking about structure, RNA has single strand that can fold into complex shapes. Whereas in DNA, double strand, that is forming double helix, twisted double helix. Function, it carries the genetic information, as I have told you earlier, that participates in protein synthesis and form regulatory roles. And DNA, this contains the genetic information, the hereditary information, and they serve as the blueprint for life. Talking about the stability, they are less stable and degrades quickly, whereas the DNA are more stable and passes genetic information from generation to generation. So for that reason, it should be stable, because this is the main building block for our hereditary. Now, if I talk about the location that are found in the cytoplasm, nucleus, and ribosomes, whereas in case of DNA, this is found in nucleus and chromosomes. For the types of RNA, we have messenger RNA, that is called as mRNA, that carries the message. Ribosomal RNA, that carries the ribosomes. Transfer RNA, that transfers this thing. So messenger gives the message. Transfer RNA transfers the assets, the tRNA. Then is the microRNA. Talking about DNA, we have genomic RNA, mitochondrial RNA, and plasmid RNA. Sorry, DNA. So this was the difference between RNA and DNA. Now let us look at the biological function of nucleic acid. So we have mentioned it right now in the different differences also, but I'm mentioning it again, that DNA is the chemical basis of hereditary and may be regarded as the reservoir of genetic information. Reserve of genetic information. A DNA molecule is capable of self-duplication during cell division. So whenever a cell duplicates to form a new cell, the DNA also duplicates, and this thing is facilitated by the RNA. So that tRNA, mRNA, and all those RNA are doing this thing only. They are replicating our DNA. Now, other important function of nucleic acid is the protein synthesis in the cell. Actually, the protein are synthesized by various RNA in the cell, but the message for the synthesis of a particular protein is present in DNA. Clear? So this is the biological function of nucleic acid. Now next, hormones. So from your teenage time, you must have been hearing this word hormones, that hormone changes have been taking place. So let us study some things about this hormones. So what are hormones? Hormones are kind of intercellular messenger. So they are produced by endocrine glands in the body and are poured directly into the bloodstream, which transport them to the site of action. So in terms of chemical nature, some of these are steroids. So hormones, steroids is a hormone. Example, estrogen and androgen. Some are polypeptides, for example, insulin, that is responsible for your say reduction in glucose amount. Then we have endorphins and some of the other amino acid derivatives such as epinephrines and norepinephrines. Hormones have several functions in the body. They help to maintain the balance of biological activity in the body. Like we have the example of insulin that keeps a check on the blood glucose level, as I have told you earlier. So those who are suffering with diabetes and all, they are therefore given this shots of insulin in the form of injection, so that to control the glucose level in their body. So within the narrow limit is an example of this function. Insulin is released in response to the rapid release in the blood glucose level. So for those who are diabetic in nature, this insulin release is inhibited, and therefore the glucose level rises. Where on the other hand, hormones glucagon tend to increase the glucose level in the body. So this glucagon, okay, so this is the opposite of insulin. This increases the glucose level in the body. The two hormones together regulate the glucose blood in the body. So what do? They act harmonically and mutually, so that the glucose level is maintained in our body. Whereas epinephrine and norepinephrine, they mediate response to external stimuli. External stimuli means, suppose you touch something, then you get a sensation that you're touching something, or you get say burnt by some fire, or say something hot thing, then you get a sensation of heating, and therefore your body stimulates or reacts in say action to that. So that stimulation is being regulated by these two hormones. Then we have the growth hormones and sex hormones that play in growth and development. So that pituitary gland that releases a special hormone that is important for our growth, means for height and all. Then we have the thyroxine that is produced in the thyroid gland. So our thyroid gland releases thyroxine, and it is an iodinated derivative of the amino acid, that is tyrosine. Now this thyroid gland releases thyroxine, and it is important for our body because the disease that is termed as goiter often develops for those who lack iodine. So abnormally low level of thyroxine leads to hypothyroidism, which is characterized by letharginess and obesity. Increased level of thyroxine causes hyperthyroidism, and low level of iodine in the diet may lead to hypothyroidism and enlargement of the thyroid gland, and this situation is called as goiter, Goitre. So that for that reason we consume iodized salt. So this condition is largely being controlled by adding sodium iodide to commercially table salt, that is the iodized salt. Clear? So this was for today's lecture. So what we have seen today, so we have seen enzymes. We started from enzyme. We have seen that how is enzyme important, how they catalyze the reaction, and then we move on to the mechanism of the enzyme. So how this log key model that is being deployed for explaining the mechanism of your say enzyme, in which the log was substrate and your key was say your enzyme. Now after the reaction, you get your product back from the substrate, but you get the enzyme concentration back, means the mole which was consumed in the formation of the product is given back. And one more thing, 2-3 points I want to mention about enzyme. First one is that enzyme are specific in nature. Specific in nature means they act upon a specific substrate. Second is what? Enzymes are efficient, means they worked efficiently only when there is a temperature, certain temperature, I would say, not a temperature. There is a certain temperature, which is called as optimum temperature. So when did you observe this, that your enzyme only works at optimum temperature? So you must have seen that whenever you get sick or you get bed rest, then your body temperature rises, and that is measured with the help of thermometer. Now, what happens due to increase in body temperature? So as the body temperature increases, the functioning of the enzymes inside your body that gets affected, and your body regulation changes. So due to this reason, the increase in body temperature is thought as an indication that your body is not healthy. Further, enzymes work at a given pH. What is the meaning of this? Means they work only at a given pH. How can we see this? So you must have say experienced acidity and all. So why does it take place? So because the pH that is inside your stomach is being changed due to unhygienic food, or say improper timing of food. Due to that reason, suppose in the morning you are not taking your food, but your stomach is releasing HCl. Now what will happen? That HCl will result in the say increase in your acid in the body, because the HCl that was released for the conversion or breakdown of the food is not being consumed has been there in the stomach. For that reason, your body pH changes and it results in acidity and all. So therefore, doctor advises often that you must take your food early at time, okay, so that the H plus is not released or the HCl is not released and your body functions properly. So these are the three things. This pH is called optimum pH, obviously, optimum pH. So these are three important points about our enzymes. So enzymes, I would say that they are very important for our body, because each and every reaction or each and every biological activity that takes inside the body is very complex to perform, and they cannot be done easily without the help of this enzyme. Say we have hemoglobin, which is very important for our body. It is also a type of enzyme. It also contains type of enzyme, and that helps in the proper functioning of the hemoglobin. Clear? Now let us look at then we move to vitamin. As I've told you, vitamin are nothing but there's organic molecules that are important in small amounts, and some of them are, say, produced inside our body, and some of them are not produced inside our body. Like our vitamin B12 is being removed from our body in the form of urine because it is water insoluble. Okay, water soluble. So what are the examples of fat soluble? ADEK. And the example of fats water soluble is B and C. Got it? Then we move to nucleic acid. Nucleic acid, which with the examples were DNA and RNA. They are also termed as polynucleotides, because nucleic acids were nothing but a long polymer of this nucleotide that is made up of nucleotides, and they are attached by phosphorus diester bond. Now, if I talk about the chemical composition of nucleic acid, as I've told you earlier, it is made up of one pentose sugar, one phosphate, diester bond, and one base. Now, this base changes accordingly for different nucleic acid. Like for DNA, we have adenine, guanosine, cytosine, and thiamine. Whereas for RNA, we have adenine, guanine, cytosine, and uracil. Then we move to the structure of nucleic acid, that what is nucleoside and nucleotide. So nucleoside is without the phosphodiester bond, and as we introduce the phospho group, then it is called as nucleotide, and these groups attached one by one to form our, say, this long strain, which is called as RNA. Now, if there's hydrogen bonding formed between two such strains, then it results in the formation of DNA. Then we have seen that we can use this AGCT, where we have seen that A binds with T through hydrogen bonding, and two hydrogen bonds are present between one AT pair, and three hydrogen bonds are present in between one CG pair. Clear? Then we have seen the formation of DNA and the structural aspects of DNA and RNA, that what are the differences between DNA and RNA. We have seen that. Okay. Now we have seen Next, about the biological function of nucleic acid. At last, we studied how hormones are very important for our body. With this, I would end my lecture, and other theoretical part of this biomolecules is now done. Thank you.